Digital Week, brought to you by the organizers of the TIDES Conference Series visiting Amsterdam next week, Kyoto in March, and Boston in May 2024. My name is Carter Anderson. I'll be your host for today's session titled Seamless Scalability of LNPs and Other Nanoparticles, from 200 microliters to 1 billion doses in any quantity in between. First, I'll cover some quick housekeeping items. If you're experiencing difficulties with audio or advancing slides, refresh your screen with F5. If you're experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, submit your questions in the Q&A window on the left-hand side of your screen. In 24 hours, you'll receive a link to watch the recording of this session. Let's now begin by introducing our speaker, Dai Hayward, CEO of Micropore Technologies. Thank you for joining us today. Now, I'll hand it over to you to begin your presentation.
Well, I'm speaking to you from Barcelona on the eve of CPHI. So good afternoon from here, but good morning to those in the US, and good afternoon, good evening, and even good night to those from the East. I'd like to walk through Micropore Technologies' approach to LNP manufacturing and other nanoparticles, simply because we're the new kid on the block and we're different from other people. But first, a little context. The context is the company was founded in 2003, 20 years ago, and from that very beginning, we were involved with PLGA microspheres and have continued to be since then. And you'll see down the left-hand side a timeline of some of the critical events that have happened, where we took a little while to scale up the technology from 2003 to 2018, where we launched our current range of products and got into phase 1 clinical trials a bare 3.5 years later, followed by very large scale manufacturing, followed then by an expected phase 3 to follow later this year. By comparison, our entry into LNPs and nanoparticles is much, much later. Based around the pandemic, we started in 2020 when I asked Professor Yvonne Perry at Strathclyde University to take a look at our technology and see if it would work in this area. In other words, would it make nanoparticles in the same way that it made microspheres? To which her answer was, yes, it does, and does very well indeed. So since then, obviously, the pace of this has been a much faster pace than down the microsphere arm of our business. And we're in phase 1 clinical trials, just entering, and we've got somebody for whom we are building a 1,500 liters an hour manufacturing plant. So with that in mind, let's just take a look at the microspheres, just to put it in a context. You'll see here a PLGA sample where we were targeting a sample size of 25 microns with a very narrow size distribution, as represented by the graph on the right. And you can see the D10 and the D90 that illustrate just how tight that distribution is and how tight that CV is down at the bottom with 11.3%. That's the fundamental starting point of our technology, but actually it turns out to be a bit more interesting than that, because it's not only monodisperse, very nice narrow size distribution, but because you can change the size of the particles with the operating conditions, typically flow, but not only flow, you can go from large particles to small particles with ease, so that you can move that peak up and down within limits, of course. You can't go from 1 micron up to 1,000 microns with exactly the same configuration of equipment, but you can do quite a lot of variation within the scope of it. And that leads us to what is a pretty important graph here. This graph, the middle line is the D50, and it represents what happens if we simply control shear, as represented mostly by flow, but not only, as I've indicated. And the top line is, of course, the D10, and the bottom line is the D90. But what that shows is a great deal of controllability, a great deal of predictability about the management of your size against the operating conditions. And that means that engineers really quite like this, because they can look at that and say, oh, this is a control curve. I know what to do with that. I know how to manufacture my product using that in a very predictable, engineered way. And that's the starting point for our developments in nanoparticles. So let's have a look at the nanoparticle process as it is today. We've got a continuous manufacturing process, which is maybe a tea mixer, maybe a microfluidic based system. And we've got the LNP going into a bulk collection, and all of a sudden you've turned your continuous manufacturing into batch manufacturing. And you do some compounding, you do some TFF to exchange your solvent, your buffer rather, sorry, and also to reconcentrate up, so that you eventually end up with something that you can push through a sterile filtration process and into fill finish. And that's kind of where everybody is today. And we all know, those of us who've been involved in this, that actually you can take your RNA in buffer and your lipid in ethanol mixture, and you can use a couple of Eppendorfs and squirt the material into a beaker, and you'll make LNPs. The trouble is, of course, that is not a controlled process that will pass any regulatory scrutiny whatsoever. So in reality, what this is about is bringing a degree of control to any process, and being able to think of that control in a very scalable way that is suitable for manufacturing. Now, my COO and I both come from manufacturing. So we start by saying, what does manufacturing look like? And how can we scale it down? Not what can we do at the smallest scale and how can we scale it up? And that starts to bring a fundamentally different approach to this whole business of making nanoparticles. So let me introduce our technology. It's based on a membrane, and you can see that we've got what outwardly might look like a tea mixer, but it isn't. What happens is that lipids in ethanol go round the membrane, as illustrated in the cutaway, and through the membrane into the RNA buffer that's flowing through the center of the membrane. And that then gives you, through the self-assembly process that we're all familiar with, the RNA-loaded LNPs. And it does so in a very controlled way, and I'll come to the control in a second. But fundamentally, what we end up with is a line very similar to that we've already seen, represented by the top graph, where we can control by changing the flow, and we can bring the size down to what is an irreducible minimum based on the formulation itself. Compare and contrast, for example, with tea mixers, where they operate quite well at a particular flow rate, but that flow rate, if you change it, you go away from optimal mixing to a very unoptimized mixing, because you get various vortices and tornadoes and the like. So you get an issue of the ability to control being a challenge. And in fact, when I have sat in front of senior industry executives and said tea mixers or impinging jets are a bit of a nightmare from a manufacturing point of view, I usually get a lot more comments about not only that, but maintenance and downtime and so on and so forth. So there's a recognition that tea mixers did a fantastic job at getting jabs into arms during the COVID period, but actually there's a recognition that there needs to be a generation 2, an evolution of the process that allows people to think in terms of some sort of more reproducible, more controllable, more versatile process. And that's what I'm bringing today. So we've got a lot of data, too much to be able to include here, but a couple of salient points. We have here, on the left-hand side, a demonstration that under varying flow rates, we've got something that is laminar flow. So this is an in silico experiment, very precise. The empirical evidence supports this, backs it up, and we've got a very short mixing period in which we get that complete mixing. So therefore, a very quick self-assembly and a very reliable approach to the process. And that's represented when we look at the graph on the right-hand side. Here we have two different formulations where simply we have a difference in the degree of pegylation of your formulation such that you've got the ability to bring your size down based on the formulation, but there is always an irreducible minimum below which your chosen formulation will not go. We've got a nice tight PDI, and I've got plenty of examples where that PDI is around 0.1 or below. But I'm choosing this one as a, let's say, a more representative example of something that is real rather than just simply modeled. So this gives you that continuity from what we have already seen from our microsphere experiences. In other words, the technology is translating from making microspheres to nanoparticles and doing so reproducibly. Do the products work? Well, we get very high encapsulation efficiencies, so that we've got an assay here which is showing high 90s, and in fact we've got a PhD at Strathclyde University who's now regularly getting around 100% encapsulation efficiency. We've got a standard dose response curve, and of course we've got a cellular expression. So they work. And of course, depending on the formulation, you will see all of those data being generated with our customers' specific formulations rather than our example ones from Strathclyde. We're not changing the chemistry. What we are doing is changing the ability to control the chemistry. And that's the critical aspect that we're bringing to this entire exercise. So, do we have mice pictures with in vitro and in vivo work? Yes, of course we do. Do they show the same effects as everybody else's mice? Yes, they do. Am I going to show them today? No, because that isn't where I want to go. What I want to do is to start talking about manufacturing and therefore reproducibility and scale-up issues. So here are some of, and this is not an exhaustive list, but here are some of the factors that go into making an integrated process for making LNPs. It's not just the LNP formulation, but it's the stabilization, it's the TFF, in other words, the concentration and buffer exchange, and eventually, of course, the terminal sterilization. And these are just some of the issues that need to be watched and monitored and carefully managed. And what we're trying to do is get to an integrated process. So we have the ability to do some, because of that control curve, to do some PAT, process analytical technologies, on the manufacturing process. So if we can manage to assess the particle size and the PDI of the material that's coming out of the process after LNP formation, or indeed after dilution, which stabilizes the LNPs, then we're able to feed back using that control curve that I've already shown to be able to adjust the control to get you to the LNP size that you wish. And sufficiently interested in this are the FDA, that they're doing some work on this with one of our units that they've had for a couple of years. So this is really quite encouraging and quite interesting. So let's look at how this is embodied. What do we look like? What do we mean in terms of an overall manufacturing process? So this is a manufacturing unit. This is an example for now. This is capable of doing up to 2,000 litres an hour, as it says there, fully GMP compliant, Part 11 compliant, and all the rest that you would naturally expect. But it's important to recognise that what we're talking about is actually scaling down, and the unit that has moved from the manufacturing unit to our high throughput screening Pathfinder is the same unit, what we call the AXF Mini, that will, on the Pathfinder, give you 200 microliter samples into a 96 well plate. In other words, it will minimize the amount of RNA you need. But if you then take it into the Horizon, you can then go to around just under 100 million doses of your final COVID vaccine or similar. That's the only difference between those two, effectively, is the difference between having a syringe pump that will deliver relatively small quantities for lab compared with, in this instance, gear pumps or similar that will give you continuous manufacturing in a plant. So we have the Pathfinder 20, which is the basic unit I've just mentioned, which will give you a 96 well plate. And you don't have to use all 96 wells, of course. Most people manage with somewhere in the region of 20 to 30, sometimes 40. So very minimal RNA, and it will give you that control curve that we've already seen, around which means that you've got the ability to do your formulation and process development. We've got the next scale up, which is the Pathfinder Pro 250. This will do, as well as the 200 microliter sample. You'll see that the unit on the top has grown from a single length to double length. That's because we've got the dilution incorporated in this one, and hence the two extra syringes on the left-hand side giving you the diluent buffer. That allows for you to make up to a 250 ml sample, and it is GMP compliant, Part 11 compliant. So this unit will take you from your very first experiment into clinical trial quantities for phase 1. Without changing equipment, without any alterations in any way. That same single piece of equipment will take you all the way to phase 1. And then, as I've already indicated, we've got manufacturing with the Horizon system, where we can change the size of the unit. We've shown you the AXF Mini-based version already, which will give you up to 100, very nearly 100 million doses in a manufacturing environment. The dimensions of this, by the way, are 1 meter wide, 1 meter high, by about a half a meter deep. So a very small footprint. And we probably can reduce it even further because the big blue things are the flow meters that give all the control. And we're actively looking for things that will, for flow meters that will reduce that in overall size. So we've got that. And then we go to the Mini, which the Mini that we've already talked about, fully GMP compliant with DQ, IQ, OQ support, as well as, of course, PQ with the customer, and full PLC integration and everything that you would expect. And then we go to the largest size, and we can have the Horizon N. This one is illustrative of the Horizon 4, where we have 4 units, modular units, single pump input for each of the phases. And this is capable with 10 of those units of going up to 2,000 litres an hour. And it's one of these that we are supplying for a customer who we will have installed at around the end of the first quarter in 2024, a 1,500 litre per hour unit. And at the same time, we will probably, around the same time, we will be commissioning a 6 litre an hour unit with rather more manual operation for another customer, again, in the East. So that's why the title of this talk is 1 billion doses from 200 microliters to 1 billion doses and anything in between. Just to give you an indication of the fact that this is fully GMP, this illustrates the variety of cleaning protocols that are available, for example, so that you've got the ability to clean in place, you've got the ability to steam in place, you've got the ability to use pressurized hot water to similarly clean in place, and also you've got the ability to take it apart and autoclave it because it is so small. The AXF Mini, for example, is about 2 inches, 5 centimeters in length. So very small, very pocket-sized. And if I were doing this on a podium, I'd be waving it at you now. So where does that take us? Well, with the integration that we're looking at, we've got the top process that we've already described. But what we are seeking to do is to have the AXF technology embedded as a continuous process right throughout the entire manufacturing activity. So the ethanol adjustment and removal we can do with the first phase of a single pass TFF, and then its reconcentration and perhaps and the buffer replacement, and perhaps another step of the single pass TFF, and maybe a third. This is in development at the moment and is very much a work in progress. But this is where we're going, so that you have a true single pass continuous process through from the beginning right through to your vial filling, with therefore obviously the potential, with the right CQAs in place, for a full process that is enabling potentially real-time release. Now, I know there's a lot more to real-time release than that, but it starts to be steps on the way to that. And where does that take us? Where does our vision get us? Well, our vision gets us to 2 versions. The top version is to further miniaturize this for individual cell and gene therapy, individual personalized therapies, instead of shipping materials 3/4 of the way around the world and back, so that you might have a, in inverted commas, coffee machine sitting in your local hospital, able to take the sample, do the necessary, and get it back up to the patient very quickly. Alternatively, because it's such a small footprint, we have the ability to put the whole thing from DNA synthesis right through to vial finishing at the rate of about 50,000 doses a day into a single 40-foot container. And that enables you to put the 40-foot container into any of the LMICs. So you can think of this going into a hospital car park in Dar es Salaam or Nairobi and moving it on to Colombo or wherever. And we're working with CEPI on a project for this right now. So there's really quite a lot of excitement in Micropore around the potential for taking this manufacturing, this controlled, this very predictable, this very scalable, this highly engineered process, taking it out into the world in a couple of different ways. And with that, I'd like to thank you all very much indeed for listening. And there are my coordinates. And I'm more than happy to answer any questions that you would like. Just to give you an indication, I was in India a couple of weeks ago, and I had 4.5 hours of Q&A. Time won't permit that, but please, please do ask any questions you like.
Great. Thank you for an excellent presentation. We've received a few questions already, but we'll give the rest of you a moment to enter your questions in the Q&A box to the left of the slides. Before we begin the Q&A, I'd like to first thank Micropore Technologies for sponsoring this Digital Week. Now let's begin. All right, we have the first question here. Do your membranes last a long time, or are they consumables?
Real good question. They are not consumables. They last a very long time. We started developing the technology that I've shown you, the scale-up technology that I've shown you, back in 2016 to the eventual launch of the technology in early 2018. And we in the lab are still using membranes from 2016, from those very early days. They've been stood on, but they still work very, very well. We find that people are choosing their membrane replacement approach based on their internal compliance and their conversations with their regulators rather than any constraint imposed by Micropore.
Excellent. Okay, we have a question here. Could the AXF Pathfinder be placed in aseptic environment?
Yes, it can. It will sit quite nicely inside a laminar flow hood so that you've got the ability. We do actually have what we call the Pathfinder Pro 1000, which I've not shown, which has reciprocating syringe pumps, which therefore gives you larger volumes than just the 250 milliliters. Those reciprocating syringe pumps, for it to be compliant with a regulator, would need to be in an aseptic environment. And as I say, the Pathfinder will fit quite nicely in that. The overall dimensions of the Pathfinder are about 400 millimeters wide from memory, something like about 500 millimeters wide, about 300 deep, and about 400 high. So yes.
Great. All right, we have another question here. Have you already incorporated light scattering in your machines to monitor the size and PDI of the LNPs?
No, that's a real good question. We've done it so far. We tend to use DLS at the moment. We are actively looking at SACs and other light scattering possibilities. Of course, getting real-time analysis in line is not necessarily a trivial thing, particularly at the high flow rates, the high capacity units. But yes, you know, they're all under active investigation, no question.
Great. All right, we have another question. Are you considering a single-use version of Micropore mixing devices to avoid SIP/CIP hurdles?
That's a really good question. We're finding that the world is kind of dividing into two camps. Those who like single-use items and those who now prefer something that is reusable. And if I'm honest, what we're seeing is, because of ESG requirements or ESG reporting, what we're beginning to see is a trend towards multi-use and cleaning rather than single-use and throw away. It's not necessarily anything to do with the unit itself. Have we considered a single use? You can use it on a single use basis, but we're suggesting you don't need to. We find that the clean in place is perfectly adequate. The protocol that we've developed, it has been approved by a couple of regulators. So we're not seeing any major issues in there that limit the cleanability and therefore the ability to use single-use, to use multi-use equipment like the membranes and the rest of it. One thing I will mention, and we've talked here about LNPs, of course, is the fact that this is a technology, because all of the materials of construction are stainless steel. If we're looking at nanoparticles that comprise or need other solvent systems, some of the hybrid systems, for example, or polymer nanoparticles, then at that point, of course, single-use plastic systems become a bit of an issue because of leachables and extractables, and there's a conversation to be had with the regulator. Whereas of course, with 316L stainless steel, that's really a very easy conversation. So there are a variety of reasons, and we're very happy to discuss on a case-by-case basis the requirements with a customer.
Great. All right. Another question. As applied in the 40-foot container, how will these be tested for product characterization and biosafety?
That's a level of detail we've not yet got to, because at the moment this is a concept. We're looking for, we're doing some of the work with CEPI to identify the steps to be able to put in place the monitoring and the management of those CQAs that I showed earlier in the presentation. But at the same time, we recognize that there's going to be a need for other partnerships to take this forward to a full, complete, deliverable package, if you like. So there's still work to be done in that.
Great. All right. Another question. What will be the surface charge of mRNA-loaded and LNPs in neutral pH at the end of the manufacturing?
I have to confess, I don't know the answer to that one. That's usually left to our customers to talk about, because at the end of the day, these are formulation-specific issues, and we're formulation agnostic. So we work with customers who come with their own lipid mixes to achieve their own specific objectives. We can help with the formulation development, of course, but in overall terms, that's something that they concern themselves with rather than us. So I'm sorry, I'm unable to answer that question.
Okay, another question. Can we adjust the pore size of the cross-flow membrane?
Yes, of course we can. The smallest we can go is to 3 microns. Typically, we find that 5 or 10 is the sweet spots for LNPs. But of course, if we're going back to the microsphere conversation, then we can go up in size. And in fact, the largest we've ever done is about 350 microns in size for very large microspheres. But yes, we can not only engineer the size, we can engineer the shape, we can engineer the spacing, and we can engineer the pattern. So it might be a diamond pattern, it might be a square pattern, and we find that all of these make a difference to the way that a customer's formulation can be optimized to give them precisely what they want. So we work very closely with our customers to make sure that they get the optimized version of their product based on our technology.
Excellent. All right, another question. Have you worked on minimum shear stress required to get LNPs?
That's a question that's an interesting one. We do have some data on that. It's a work in progress at the moment because we're trying to validate and reassure ourselves about that data. If I say that the operating pressure under which our systems work is typically between 2 and 5 bar, and might go in an extreme case as high as 10, then you will see that that's very, very much lower than a typical microfluidics or a typical T-mixer environment, where typically they're in the region of 20, 30, 60 bar pressure. So the shear forces involved in what we're doing are very, very much less. We do expect to produce a publication on this fairly soon, however.
Okay, another question. Can your engineering technology help stabilize LNPs for continuous administration to patients without low temperature requirement?
This is an ambient temperature process. So in principle, the answer would be yes. And if we are able to get to the point where we've kind of got the coffee cup, the coffee machine manufacturing device, then the answer to that question will be highly likely to be yes. We're, again, a bit like the container, we're some way off that at the moment because we need to develop the inline processes, the continuous processes, and the analytics that support that so that we can be sure that that is directly administrable to a patient. In theory, the answer will be yes, but we're some way off that.
Great. Okay, another question. Thank you. And another question is that 200 microliter minute machine supports inline concentration, or we may still need to use Micropore to concentrate the products?
At the moment, we can do the dilution at the 200 microliter sample size, and it's not 200 microliters a minute, it's 200 microliter sample size. As far as the downstream is concerned, after the stabilization stroke ethanol dilution, then that at the moment is a batch process of TFF. Now, we all know that the TFF process downstream, as given by TFF or dialysis at the small scale, is it's a destructive process. It's where most of the problems arise with manufacturing these materials, these LNPs. And one of the things we're aiming to do when we do produce the single pass version is significantly to reduce that destruction because of the low shear involved in our continuous process. So at the moment, we do not have that next step. The plan is that we will by the end of the CEPI the CEPI process.
Great. Okay. What is the mechanism of action of LNP formation in AFX systems? How is it different than T-mixer?
It's fundamentally the process of LNP formation, in a sense, at the simplest level. And of course, there is, an academic debate about just exactly how these are formed and what their final constituency looks like, and so on and so forth. But for the purposes of this conversation here, as I've indicated, 2 Eppendorfs into a beaker will make you LNPs. So in principle, any way of mixing your RNA with your lipid will give you LNPs. The challenge is less how do you make LNPs, because they self-assemble. The question is much more, how do you impart control over the manufacture of LNPs, because that is far more variable. And as I indicated on one of my slides, LNPs made by the T-mixer approach, while ostensibly really quite simple, actually are very difficult to go from one scale to the next. I actually heard somebody at a conference in June from one of the major manufacturers talking about, in answer to a question, how do you scale your process? The answer was guesswork, which I find really quite fascinating. But it was indicative that going from one scale of T-mixer to another needs a lot of revalidation whereas I've indicated already that our approach doesn't need that revalidation because the same device will go from that 200 microliter sample up to the 80, 90 million doses of product. So from that point of view, T-mixers will make LNPs. And, you know, we all know it. Pfizer are well known for the videos that they've got on YouTube and elsewhere around the way they've used T-mixers to make their vaccine that got jabs in arms real quick. And I take my hat off to them. They did a fantastic job. And I'm not going to say anything bad against them. However, in conversations with Pfizer, they recognize that there is a need for a second generation approach to this that gives them more control, more reproducibility with a lot less maintenance. And that's the thing that we're bringing to the party.
Great. Okay, we have a few more questions here. Please, can you give a little more detail about how you're able to scale up?
Yeah, we basically, what we've done, as I say, from a manufacturing point of view, when you are starting in the lab and go from lab to manufacturing, particularly in a continuous process, you need to keep certain things as constant as you possibly can. And we're all familiar, those of us particularly who've been in a manufacturing environment and received a process, a chemistry process, which had in the lab a minor exotherm that wasn't really noticed, where that can be a runaway and take the roof off your plant. So getting to a place where you understand the fundamentals of the process and therefore can scale it because you understand things like keeping the geometry the same, keeping the Reynolds numbers the same, giving you the same shear force. Issues of that sort give you the ability to control the way you scale. And that's where our fundamental approach to scaling comes from. We start from that manufacturing background, we start from a continuous manufacturing background, and we look at what are the things we need to be able to control all the way through from the smallest example, the smallest sample, all the way through up to the largest you could possibly ever want to go. And that's what we've been able to do with the geometry and the configuration and the engineering that we've applied.
Excellent. All right, looks like we have our final question. What help do you offer companies on GMP?
My COO comes from a GMP manufacturing background. He used to work at Fujifilm Diosynth. So he knows and understands the rigors, the questions, the issues associated with GMP. So we start at the very beginning with the idea that this is going to end up as a CGMP process with any of our customers whenever we start to do some work. So we can start by working in our own labs to do formulation and process development in the way that I've already described. We then work through a technology transfer process, which is fairly simple, fairly straightforward, because of the scalability that we've talked about already. And that enables us to give the support right through from the formulation development, through the manufacturing issues of DQ, IQ, OQ as a part of the total package. We understand Part 11 compliance very well, so it's all part of that. We understand cleaning and sanitization and sterilization and what's required there to satisfy a regulator and how different regulators behave around the world, because they do, and so on. So we're there, and then we will work with the customer, with our clients, during their PQ process, which of course is theirs, but we're not hands-off at that point. And in fact, we continue to support our clients through all of their subsequent developments during their process, if you like, and their manufacturing for their product lifetime.
Great. And it looks like we actually just had another question coming in. I would like to know about the simple routes to prepare LNPs.
That's an interesting question. I mean, I think I've already said the simplest is a couple of Eppendorfs with one loaded with RNA and buffer, one loaded with your chosen LNPs, and squirt them into a beaker. You'll make LNPs. That's the simplest. It doesn't get any simpler than that, really. So you can make LNPs that way. Does that give you anything you can work with to start to build a dossier in terms of physical characteristics, in vitro results, and eventually, of course, in vivo results? I would argue it does not. I would argue that it is not reproducible. And although you've made LNPs in seemingly a simple way, you've done so in a way that doesn't advance your cause. There are other ways. Of course, we've talked about microfluidics. We've talked about T-mixers. And there's us as an alternative. We sit between the two. All three are perfectly viable methods of manufacturing LNPs. It just depends what your agenda is about what you want out of the stage you're at any particular point. And of course, you know, if you're starting with something on the bench that you may then, as we've described with T-mixers, need to go through a process of revalidation as you scale up, then that adds time, it adds cost, it adds complexity, and it obviously adds vulnerability to your arguments with your regulator about have you done everything you thought you should have done, or that they think you should have done. And if the answer is no, go back and start again, or do something extra, that adds further time and cost. The difference with us is we just simply bring something that'll get you from the beginning to the end in a very predictable, scalable, proven way. And by the way, it is simple enough that a couple of years ago, my 14-year-old grandson, with half a day's training, was able to make perfectly good LNPs using this technology, albeit in a simpler way, an earlier stage of its development. But he, as a non-scientist, was able to produce robust, reproducible LNPs with half a day's work.
Great. Well, that's all the questions we have for today. It looks like we're about 4 hours short of your Q&A session in India, but thank you, Dai, for a fantastic session.
Thank you very much, everybody.
Great. And yes, this session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. Before you log off, please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Informa Connect Life Sciences, have a great day.